Okay, the existence of CD8 T-cells is to fight off any organism that has the ability to infect a cell. What's the advantage for an organism to get inside a cell? There's two main advantages. Two, sorry? Okay, viruses replicate, they have to replicate, they're intracellular. That's one thing. Okay, so three main things then. What's the other one?
Nutrients.
Loads of nutrients in a cell. If you're inside the cell, you just suck on that shit, you know? Literally, it's wicked. Okay, that's the other advantage. And the third advantage, exactly, you can hide. Okay, is it better to be an extracellular pathogen or an intracellular pathogen in terms of host defenses and stuff? Intra, because inside you only get like T cells, CD8 T cells, and NK cells that can kill you, right? If you're in, I don't know, the blood or something, you have all the blood molecules, you have loads and loads of antimicrobial stuff in there, you have complement, you have everything that can attack you. If you're inside a cell, you're in like kind of like a little cave, you can just protect yourself. So it's really good to be inside a cell. So that's the advantage of bacteria and stuff like that. Okay, I talked about T-cell development. So T-cells from the bone marrow, they go into the thymus, they mature in the thymus. Okay, this is where clonal deletion comes into sort of effect. The first mechanism of clonal deletion, that is, in the T-cells, in the thymus, if let's say a T-cell recognizes anything, it will commit suicide. Okay? Does that make sense to everyone? Okay? If the T-cell, in its developmental stage, recognizes any sort of anything in the thymus, it'll kill itself, because the thymus should be a sterile area. It should only contain self cells. So if a T-cell is recognizing a self cell, it has to kill itself, because that means it's an autoreactive T-cell. Does that make sense now? Yes? Okay, so that's a bit about the development. I said last week that there's two main types. There's alpha beta and there's gamma delta. Alpha beta are like 95% of the time the most common type of T cells. So in alpha beta T cells, you have two types. You've got CD4 and CD8 T cells. And they're called CD4 and CD8 because they have a CD4 or a CD8 molecule on their cell surface, one or the other. Right, gamma delta T cells aren't so important. Dan loves them, so ask him about them. Okay. Okay, CD8 T cells kill stuff by apoptosis. What's the difference between apoptosis and necrosis?
Apoptosis keeps it contained within the cell so that you don't get all the damaging factors spilling out into the bloodstream.
Exactly. Sorry?
I was going to say that necrosis is automatically what happens on the cell's own investigation. Necrosis can be on the cell.
Exactly. And what did you have to say?
Program, so it's active. Necrosis is non-program.
Repeat that again. I didn't hear that.
Apoptosis is program, said there, and necrosis is non-program.
Yeah. Sorry, yeah, you're right. Okay, so let's combine all that stuff together. All right, I'm not going to ask you any more questions now. Apoptosis versus necrosis. Apoptosis is controlled programmed cell death, right? You're doing it so that basically whatever you're trying to kill, it's localized to what you're trying to kill. Necrosis means literally you're just killing everything in the nearby area. You're sort of all the contents are spilling into the blood, and it's causing mass destruction. It's pathogenic. So if you have a heart attack, you get necrosis of cardiac muscle. Okay. Apoptosis. My example is like you get a tadpole, right, and its tail, when it becomes a frog, the tail undergoes apoptosis over time. So it's programmed, it's controlled to that area. It's just the tail that's dying, and then it becomes a frog. Okay? That's apoptosis versus necrosis. Apoptosis is what's used by CD8 T cells to kill stuff. There's 3 molecules, not the 2 that he mentioned. There's granzyme, there's perforin, and there's another one called granular lysine. So I'll talk about that in a minute. I'm going to go through now the whole pathway of how everything happens on the computer. So have a look at this. Can everyone see that? I'm going to make it bigger. Okay, guys, so this is what I was talking about before. You get injury and infection. It's detected by the skin macrophage or something. So resident macrophages underneath the skin detect it. They release, the main things are tumor necrosis factor alpha and IL-1. That results in acute inflammation and also the mast cells and stuff. They do all the histamine and all of that stuff. Eventually, it leads to acute inflammation. If it's not controlled, then you get drainage into the lymphatic system. Okay? And eventually, what happens is when it gets drained into the lymphatic system, it will get activated. So you get... I'm trying to think of the best way to explain this. Okay, so I said in the lymph nodes you have T-cells, okay? The dendritic cells, which are part of the innate system, they've already phagocytosed some of whatever's causing that inflammation. They bring that via, when they get drained into lymph nodes, they take it to the lymph nodes, and these stuff, so an activated dendritic cell, then goes on to stimulate a T-cell. Does that make sense? That's just a summary of what we were talking about before. Now we go on to the more difficult stuff, okay? Yeah, this is why I said to come to a lecture, because you're not going to get this otherwise. It's just me having a bit of a mental breakdown one day and drawing this. Okay, so you start off in the bone marrow as pluripotent bone marrow stem cells. They differentiate down the lymphoid lineage, so they become lymphocyte precursors. Now they have the choice of becoming either a T cell or a B cell. Okay? T cells, as I've said all of this before, they migrate to the thymus, and you get T cell precursors in the thymus. They're called thymocytes when they're in the thymus. In the thymus, what I mean by maturation development is the T cell in the thymus starts off as a cell. It doesn't have any receptors on its cell surface. What happens? So it's double negative. It doesn't contain CD4 or CD8. Then what happens is they become double positive. So they have both CD4 and CD8. And then eventually they develop into either CD4 or CD8. Those are details that are not really needed, but it allows you to appreciate what I mean by development. So does that make sense? Okay, that's what this stuff is if you're reading it. Okay, so you have the T cells. Once they've finished developing, they're called mature naive T cells. Really important term, mature, naive, because they're still virgin T cells. They haven't been stimulated by anything. And T cells, that's what he refers to in his lectures as Th0 cells, Th0 cells. Okay? These cells now migrate into the lymph nodes. Okay? This is how they do it. You don't need to know, but it's there if you want to know. They migrate into the lymph nodes, and now we've sort of split it up into two categories. They've already been split up into CD4 or CD8 in the thymus. Now I'm just talking about them as two separate pathways. So I'm going to start off with the CD8 pathway. So CD8 is to destroy stuff that's been infected or is a tumor cell. So you need to know the names of some things that can infect a cell. So start off with viruses. All viruses infect cells. So the CD8 cells will then kill infected cells, right? The other thing is some certain bacteria are intracellular. The main two that you want to remember, okay, is TB. TB infects macrophages, okay? It goes into macrophages and it grows inside the macrophages. So it's intracellular inside a macrophage. So that's TB. And the other one that you want to know is leprosy. That's part of TB. TB causes leprosy. Okay, so you've got Mycobacterium tuberculosis and you've got Mycobacterium leprae. So it's the same TB pathogen. Okay, so you've got TB, and the other one that I always talk about is, where's the other one? Chlamydia. Chlamydia is really important intracellular pathogen. It's an atypical bacteria. It's a bit dodgy. Okay, you'll learn about it this week probably in your microbial lectures. It's a bit dodgy, but it's an acellular. It produces inside cells. Okay, it's intracellular. Okay, so I said to you that the first antigen presenting cell is a dendritic cell. So an activated dendritic cell, so this is where the star is on this diagram, it basically means it's joined from that other diagram I showed you a minute ago, the acute inflammation one. So it joins on. Okay, so an activated dendritic cell is any dendritic cell that's basically, it's got something inside it. So it's got an intracellular pathogen. It's been infected. Let's say it's a virus. It's been infected by a virus. Okay, and basically it's expressed all of that stuff on an HLA-1 molecule. So let's get a piece of chalk. I talked about this last week. This is the CD8 pathway, so it's all about getting infected. It's not about phagocytosis. Okay, you have a virus, it infects a cell, right? You get processing via the nucleus and whatever. You get formation of HLA-1. Yes? Virus is broken down into little peptides. One of these peptides is bound in the HLA. So I said HLA looks a bit like this, and this is the bound peptide. That goes off to the cell surface, and you get expression of a virus particle, a virus protein, on the cell surface membrane of this dendritic cell on an HLA-1. Do you remember that? Yes? Does that make sense? Or have I gone too fast?
You say it all the time.
Yeah. Virus infects a cell, so that's a virus, infects the dendritic cell. Okay? Dendritic cell basically processes the virus. So what happens is let's say you're talking about HIV. When HIV infects a cell, little bits of HIV are inside the cell. You know, they get cleaved in order to release the DNA from the capsid and all of that, right? So when it gets cleaved, little peptide fragments, these are called oligopeptide. Oligo means few. Little oligopeptide fragments are taken, so one of them is taken and put on an HLA-1 molecule. And that's basically, it moves towards the cell surface and it gets expressed on the cell surface on HLA-1. Does that make sense? Okay. Now, this dendritic cell is activated. It now goes to a CD8 cell. It presents this to a CD8 cell. So let's say this is CD8 T-cell. It's got a CD8 molecule on it. It's got a T-cell receptor. So, I told you last week that it's a CD8 or CD4, whatever it is, that recognizes the class of HLA. HLA-1 is always and only recognized by CD8. Really important that. So it's this CD8 which recognizes what class the bracket is. CD8 HLA-1. Okay? A T-cell receptor then recognizes the bound antigen peptide, whatever it is. Okay? When it does that, this is the first antigen presenting cell. It's a dendritic cell. This dendritic cell will cause the CD8 cell to become activated. Okay? So this dendritic cell causes this CD8 cell to become activated. What do I mean by activated? I said it last week. What's the meaning of activated? Increased potency. It means it's a killing weapon. It's like it's a machine that can kill literally anything.